Of course, thanks to the other speakers. I think the science part has been really great. And now I want to talk a bit about how we look at these things and these big data challenges that come with volume electron microscopy. And I really want to start with this chart, which is a big motivation for us at Scalable Minds to work on these things. And you can see here that there's this blue line, and it has annotated a couple of data sets that have been published by the connectomics community in recent years. There's also some speculation about the future in here. And you can see that this is an exponential growth because the y-axis is logarithmic. So these are the data set sizes, so they're really growing exponentially at the moment. We have this other line here, which is this dashed line, and it shows what like a single workstation computer can store and process. And the problem here is that traditional software is built to work on a single computer. And so that's really why we believe that a new generation of software tools is required to actually make these large data sets handleable and to be able to work with them effectively. So that's the first motivation and trend. And then the second one is AI. Everybody talks about it, but the reality is that it has become a pretty mature tool in the bioimage analysis field, and many people get really good results out of it, but still it is still quite complicated to apply, especially for subject matter experts. And I think there's still some opportunity to make it much easier to use. And then the third trend is that data sharing and collaboration is really taking up. It has been for a number of years now, but it's still increasing, and that's great for the science part. It's great to also disseminate the data, and other people can work on that. It's also required by some journals and funders, but it also brings some challenges with it, especially if we're talking about very large data sets. So let's maybe talk about like a specific example. Like just imagine you have this new volume electron microscopy data set. It's around 5 terabytes in size, and you want to share that with a collaborator, right? So you can buy a hard drive and put it on there. Hard drives now have like 20 terabytes or something, so that wouldn't be a problem. Then you can just ship it out to somebody. That's fine if you have like one collaborator or two. If you have 20 or even 100, then this becomes a logistical nightmare. You can also upload it to your server, and that's great because then other people can download that and work with that, so you don't have to do shipping and stuff like that. So that's already much better. But still, if then the person on the other side wants to work with that data, basically they have to download the entire thing if you're not careful about the file format, and until they actually can start working with that data, right? And that's fine if you have a big workstation computer that can fit 5 terabytes, but on my laptop, 5 terabytes wouldn't fit. And so that's another bottleneck in this process. And then, of course, with large data sets, then analysis is another nightmare for some, and really needs a new way of working with that. So what we've developed is WEBKNOSSOS, and we developed that in collaboration with the Max Planck Institute for Brain Research. And it's an open source tool for working with very large image data sets in a collaborative manner. And as the name suggests, it's web-based, so you can open up a browser and go to webknoss.org. And here's a demo data set. This is a fruit fly brain that was imaged by a group around Janelia and published a couple of years ago. And it's a 50 terabyte data set. You can just open it up in your browser, even on your iPad or phone if you wanted to. And you can zoom in and out and it's fast. Like this video here is in real time. And it's all in your browser. You don't need to install anything. And also because it is on the web, it's also very easy to share with others, right? So you can just take the URL from the address bar here and send it to somebody on your favorite chat platform, and they will be able to look at the exact same screen as you are. Of course, WEBKNOSSOS also has capabilities for working with private data. You might not want to share all of your data when uploading it for the first time. So WEBKNOSSOS is also a data management tool for these large images where you can upload your data set, you can organize them in folders, attach tags and metadata and all of that good stuff. You can assign permissions to individual data sets, who gets to see what and who gets to annotate them. And speaking of annotation, that is really the second big feature of WEBKNOSSOS apart from data sharing and visualization. And WEBKNOSSOS has a number of different annotation tools. Here you can see the volume annotation tools, and what you're seeing here is the quick select feature, which is a semi-automatic annotation tool where you basically draw a square over the feature that you want segmented, and then there's an AI model running to fill out that feature for you. Another feature that you can see here is a volume interpolation, and that, because WEBKNOSSOS was developed for 3D data, came quite naturally to us. And what the volume interpolation allows you to do is you annotate on the section, then you skip a few, right? What we're doing right here, and then it will just fill up the space in between for you, and then you can go in and correct if you wanted to. And again, everything is happening in a browser. You don't need to install anything. You can send it out to other folks to help you with the annotation work, and it's quite fast. And then you get these nice mesh representations that we can see here in this 3D viewport, and it's going to come back in a second. And there we go. Yeah, and actually the quick select feature got a lot smarter last year. Now you can just do a single click. You don't have to draw a square anymore, and it can work in 3D, right? So here we're just clicking once, and then it tracks along a number of sections. You can define how many, and it will track this object along the way. And when we introduced this feature into WEBKNOSSOS, we were quite blown away how well it worked, given that we use the segment anything model under the hood, which has not been trained on EM data, but has been trained on cats and dogs and cars and everything, but it still works amazingly well. And I can only really encourage all of you to try it out on your data and see if it works well. And if it doesn't work well, then still WEBKNOSSOS has all of the other annotation tools like a brush and your eraser at hand, so you can do corrections in there. There are also other annotation features in WEBKNOSSOS, and some have been mentioned today, like the skeleton annotation tool, which is really quick for drawing line graphs. If you have neurons, that's great, because then you can approximate them quite nicely with these skeletons. And because WEBKNOSSOS runs in a browser, it also runs on devices that have a browser, like an iPad. And there's pen support, so you can use a pen and draw if you want to, and if you want to get really precise and be efficient about that. And once you've done all the hard work of doing the annotation, WEBKNOSSOS also has some capability for exporting segment statistics based on the annotation that you've done. And then, of course, you can export that and put that into whatever analysis tool that you want to use. And yeah, that's a good way of starting your scientific analysis based on the segmentation that you've done inside of WEBKNOSSOS. Another product of your segmentation are these meshes. We've already had a brief look at them. They can be a bit more complex than just this ball or this nucleus that we were looking at earlier. You can look at them inside of WEBKNOSSOS, but you can also export them and then, for example, put them in Blender to make nice animations for your publication or your talk. WEBKNOSSOS also has an animation feature built in, so if you're happy with the presets that WEBKNOSSOS offers, then you can get an animation like this very quickly from WEBKNOSSOS, and that can be useful again for some presentations that you want to do. Specialized for connectomics, WEBKNOSSOS has a number of features. One of them is the proofreading tool, which allows users to be very efficiently in splitting and merging these segments. So here we're doing a merge, so we're clicking on these two partners, clicking merge, and then boom, now they're merged together. It also allows for split operations, what we're showing here. So there's a false merge, and then we can tell them, okay, please split it at that location, and then WEBKNOSSOS will split that for you. So, and both operations work either in this 3D view mode or in this sectional view mode as well. Another feature that WEBKNOSSOS has is working with multiple modalities. And WEBKNOSSOS has the ability to correlate images from different modalities on top of each other. So if you have a CLEM workflow, and you can upload both the LM data as well as the M data, and then correlate them inside of WEBKNOSSOS or import big warp CSVs, and then WEBKNOSSOS is able to visualize them in one scene. Here, these are neither light microscopy nor EM, but it's two X-ray data sets, one from a lab source and one from a synchrotron. But you can see that you can overlay them nicely, and then on top of that, you can also then do the annotation work just as you would with normal data sets. And this feature is still a bit experimental, so if you want to try that out, then please do. And if you find something that we should add, then please let us know. Speaking of multiple modalities, WEBKNOSSOS also has support for time series. So some of our users have these crazy large time series light sheet data sets and also upload them to WEBKNOSSOS, and again use all of the annotation features and sharing features. So that works as well. So WEBKNOSSOS is not just for EM, but can also support a wide range of other modalities as well. And because WEBKNOSSOS is a web-based tool and sits on a server, everything has an API, and that's great if you know a bit of coding, and then you can access all of that data through, for example, our Python library. So we have this WEBKNOSSOS Python library that you can use to access data set that has been stored on WEBKNOSSOS, and then you can download that data partially and do your analysis on top of that. You can, of course, also upload data. So if you have an automated pipeline, then you can use this library to just upload the data and set all of the permissions and all of that stuff. So that's really great for automating stuff. And all of this is powered under the hood by this file format called OmiZar, and Kales also mentioned that in his talk. So OmiZar is really a next generation file format that is just really great for volume electron microscopy. And if you haven't heard of that before, then I would highly recommend you all checking that out. So what it does is, like, if you look at the way traditionally these files come to be from a microscope, right? So you have your sample, you put it in a microscope, you get out these sectional images. And either this is like a TIFF stack, or it's a single big TIFF image. And then now you have that, but the way these files are stored is not really optimal for some of the use cases that you might want to do, like interactive visualization with virtual cross-cutting, or even three-dimensional processing on an HPC or something like that. So it's really not well designed for that. For one, because most of the time you need to either import the entire data set into main memory, and with 120 gigabytes, that might be fine. Some computers have that much memory, but a regular laptop doesn't. Or you have the data oriented in sections where you can only load individual sections, and that really is a bottleneck if you want to do 3D analysis. So what OmiZar provides is to organize your data in a different way. And it has a couple nice properties. First of all, you can chop it up into cubes, which is much more natural if you're working with three-dimensional data. And if you have this chopped up into these cubes, then you can access individual cubes individually, right? So you don't have to import the entire thing, but you can go piece by piece. And this enables use cases like interactive streaming that we've been seeing in WEBKNOSSOS. And so the data can live on a server somewhere, and then it can be streamed into a bunch of different tools. So here on this iPad, you can see WEBKNOSSOS. This is NeuroGlancer, which is a tool developed by Google, which is also a 3D visualization tool. And this is Mobi, which is a Fiji plugin. And all of these are compatible with OmiZar. And that's another great property of OmiZar, is that it's actually a file format that has a broad community support. So most of the open source tool developers have either an existing implementation of OmiZar or are working on one. So that's really a file format to watch. And if you want to try it out, one of the best ways of doing that is you can just go to webknoss.org and upload the data in the format that you have, and then it will automatically be converted into OmiZar, and then you can use it from there. All right. Next, I want to talk a bit about some of the segmentation services that we provide, and they've been popping up in some of the talks before. So I won't go too much into detail, but basically there is this workflow, and there are a couple of the steps that we can help you with. So we can't really help you with acquiring the images on a microscope, but once you have some images, then we can help you align those, segment those. Proofreading is possible with WEBKNOSSOS. Then you can do your analysis, and once you've published, then you can use WEBKNOSSOS also to share the data with the world, right? So you can just put in a link to your data from WEBKNOSSOS into your paper, and then use that to share that with the world. So what that looks like in detail is, at the beginning of this pipeline is alignment, and that's super important for volume EM data, because if you have really good alignment, then automatic segmentation is quite easy. And if you don't, then it's virtually impossible. So it's really useful to spend quite some time on getting the alignment right. And and we have good pipelines now for dealing with that. And then the next step in the pipeline is, at least if you're working with connectomics data, is getting some neuron segmentations in. And here also we have a pretty robust pipeline that has seen a large number of data sets by now and is pretty robust. And we've been working this on for almost 7 or 8 years now, also in collaboration with the Max Planck Institute for Brain Research, to really fine-tune these model architectures so that they are robust, not just on a single data set, but can scale across multiple data sets. And then if you want to do connectomics, you need synapses, so we have synapse detection, and some people might be interested in other features as well, like nuclei or blood vessel detections. It's of course also something that you can automatically get out of these data sets, and or organelles like mitochondria as well. So all of this is basically we have a pipeline for, and provide this as a service for our customers. And now I want to give you a sneak peek of what we're working on next, because we're actually interested in bringing these services as features into WEBKNOSSOS. And we started out with the stack alignment, and that's actually already live as an experimental feature on webknossos.org. So you can upload your EM data set unaligned, and if it's a single tile data set, then you can just click on this AI analysis button, and it will trigger a alignment job. You don't have to worry about getting the compute ready or anything about that. So it's really, you click a button in your browser and then it will do that, and then you will get an aligned data set. And there are options for fine-tuning that after the fact with some fiducials like Luke mentioned. So this is already available, and we're also going to have support for multi-tile alignments later this year. Then the next feature that we're working on is bringing automated segmentations into WEBKNOSSOS, and also already today as a preview feature, there are some pre-trained models that you can run on your data set. Most prominently for neuron segmentation. So if you have an EM data set of some brain tissue, then again, you can upload it to WEBKNOSSOS and you can segment out a portion of that data set. And you don't have to worry about buying GPUs or configuring CUDA or anything. Just, again, you upload your data, you draw a bounding box of the region that you want segmented, click a button, and then you might need to wait a bit until the data is ready. And then you can inspect that, and from there you can, and of course, download the data if you wanted to, or further analyze it or proofread it inside of WEBKNOSSOS. These are pre-trained models. You can also see here that we're working on not just neuron segmentations, but also mitochondria and nuclei, and we have a few others on our roadmap that we want to bring into WEBKNOSSOS. But pre-trained models are great and everything, but sometimes you want to really get the best quality out of your data or out of your segmentation. And for that, fine-tuning or retraining is really necessary. And doing that on WEBKNOSSOS should also then be a possibility, and WEBKNOSSOS actually quite well equipped to do that because of all of the annotation features are already present inside of WEBKNOSSOS. And if you've uploaded your data set, then you can basically run a workflow like you upload your data, you generate a segmentation with a pre-trained model, then you go in with the annotation tools, fix a few bounding boxes, and now you have high quality training data that you can then feed into this dialog again and tell WEBKNOSSOS to train a model for you. And then again, we can hide all of the complexities of running and training models for you, so you don't have to worry about that, and train a model. And then once it's done, you can roll that out onto your entire data set. And then you get a result that might look something like this. You can see here we have support for different mappings. We're working with supervoxels. That's not important right now, but what you can see here is that you can then also, again, edit this and, for example, use the proofreading tools in order to really then do like the final touches on your segmentation to get it to a state where you're really happy with, and to do your analysis on top of that. So this is really what we're working on at the moment, and if you're interested in trying that out, then please reach out to us, and if you have some ideas of what we should do next, that's of course also very, very useful for us. So to summarize, WEBKNOSSOS is really a comprehensive platform for working with volume electron microscopy data. You get your data from the microscope, then you can align it in WEBKNOSSOS, you can visualize it, of course, you can collaborate quite easily with people all around the globe because it's all on the internet. You can do your annotation work, you can generate segmentations, you can proofread segmentations. It helps you a bit with getting some data for your analysis, and that hopefully leads to some insights that you can then publish. And there again, WEBKNOSSOS can be useful for sharing the data with the wider scientific community. And on top of that, has features for dataset management. It works with not just EM data, but other modalities as well. It's all built on top of OMISR, which is a standardized file format, and that helps with interoperability with other tools as well. And WEBKNOSSOS is also cloud ready, so it works in cloud environment, but it also can be deployed on institutional HPC systems and use the storage and processing capabilities there. So if this all sounds great, then you can go to webknoss.org and create a free account. Free accounts come with 50 gigabytes of storage, so you can really upload your data, try it out, some of the features I've been showing you today. And yeah, we're really happy to hear any feedback. If you have some, you can find us on Mastodon and Blue Sky, or send us emails. And now I'd really like to thank you all for your attention. I would like to thank again the other speakers and really all of our users who really help us shape the roadmap of WEBKNOSSOS. And we really love collaborating with all of you. Thanks.